So now we are going to discuss a topic called autoimmunity. So before going to that, so first going in detail into this topic. First, we have to know what is the definition of autoimmunity. Autoimmunity is nothing but, in general, our body's immune system has the capability to differentiate between self and non-self antigens. But in case of autoimmunity, what will happen in our body's immune system will fail to differentiate between self and non-self antigens, and thereby it will consider our body's cells as foreign particles or as antigens, and shows and starts immune reaction and causes our host cells damage. So that we call it as autoimmunity. Okay, it is defined as a state in which the body's immune system fails to distinguish between self and non-self antigens and react by formation of auto-antibodies against one's own antigens. So it will consider our own cells as a foreign particles and will start producing antibodies, auto-antibodies, and causes the damage, or it causes damage to the own tissue. And in other words, there is loss of tolerance to one's own tissue. So what do you mean by tolerance? Immune tolerance is a phenomenon, like which is present even from the childhood, like even from the fetal stage, and is defined as the ability of an individual to recognize self tissue and antigens. So immune tolerance is ability of our body to differentiate, to identify what are the self tissues and what is the antigens. That is called as immune tolerance. So autoimmunity is completely opposite to this immune tolerance. And then coming to criteria for autoimmune diseases. So what are the criteria for autoimmune diseases? It includes four. They are, so the autoimmune response must be regularly associated with disease. So whatever the autoimmune response is there, it should directly involve in the disease. And then the antigen responsible for the immune response must be identified, isolated, and characterized. The antigen responsible for the immune response must be identified, isolated, and characterized. And passive transfer of the disease must be possible by transfer of antibodies or sensitized lymphocytes. So the transfer of the disease must be possible by transfer of antibodies or sensitized lymphocytes. A replica of the disease must be inducible in laboratory animals, and immunopathological changes in the natural and experimental disease should be parallel to each other. So it includes four criteria. First is it should be whatever the autoimmune response is there, that should be associated directly with the disease, and whatever the antigen that is responsible for the immune response, it has to be identified, characterized, and isolated. And passive transfer of this disease must be possible only by transfer of antibodies or sensitized lymphocytes. And a replica of the disease must be inducible, like the same kind of disease can be inducible in laboratory. So if we conduct this in vivo studies, it has to be clearly understood. So these are the four criteria for studying these autoimmune diseases. And then pathogenesis or mechanisms of autoimmunity. So the mechanisms by which the immune tolerance is broken. So when immune tolerance is broken, then autoimmunity will be taking place. So the mechanisms by which immune tolerance is broken, that causes autoimmunity. So what are the different mechanisms? Here we are studying three different factors in studying the pathogenesis of autoimmunity. It includes immunological factors, genetic factors, and microbial factors. And sometimes these three may be interacting, or any two of them may be interacting. So mainly immunological factors, genetic factors, and microbial factors. Now coming to immunological factors. So failure of immunological mechanisms of tolerance initiates immunity, autoimmunity. That means immune tolerance is ability to differentiate or identify self tissues from antigens. So if that ability, or if that immune tolerance ability is lost, it causes autoimmunity. So it involves a few mechanisms. The first is polyclonal activation of B cells. B cells are immune cells, which are mainly involved in humoral mediated immune reactions. So polyclonal activation of B cells. So what does it mean? So B cells may be directly activated by stimuli such as infection with microorganisms or any other products leading to bypassing of T cell tolerance. So when there is an antigen gets encountered to these B cells, or when antigen enters into our body, B cells get activated. So once B cells get activated, they will show these immune reactions. And also whatever the T cells are there, even these T cells also bypass the functioning of or activation of these B cells. And then generation of self-reacting B cell clones may also lead to bypassing of T cell tolerance. So T cell tolerance is nothing but T cell's ability to differentiate. That will be damaged, or that will be diverted by self-reacting B cell clones. And decreased T suppressor and increased T helper cell activity. T suppressor cells will be decreased. That means T suppressor cells helps in the suppressing of these immune reactions, and increase T helper cell activity. T helper cells will helps in the what autoimmune reactions. So if there is a T suppressor will suppress autoimmune reactions. T helper cells will enhance or increase this autoimmune reactions. So in these conditions, what will happen? There will be decrease in the T suppressor cells and increase in the T helper cell activity. So when there is more amount of T helper cell activity, it causes more amount of autoimmune reactions. Okay. And then fluctuation of anti-idiotype network control may cause failure of mechanism of immune tolerance. So any anti-idiotype network, if it is if there is any fluctuations in an anti-autobodies formation, that also cause broken, like that also cause autoimmunity. And sequestered antigen released from tissues. Sequestered antigen released from tissues, that is self antigen which is completely sequestered. Whatever our body's own tissue is there, when it undergoes complete sequestration, that means numbering, it may act as foreign antigen. So generally it's a normal tissue, but when it undergoes sequestration, it will be considered as a foreign antigen, and again, and for that again antibodies will be produced. Okay, sequestered antigen released from tissues. Whatever the normal tissue is there, when it undergoes sequestration, it will be considered as a foreign particle. For example, in trauma to the testis, if there is any injury to the testis, there is formation of anti-sperm antibodies against spermatozoa. So generally, when there is damage to this damage or any injury to this testis, our body it will consider our own sperm the individual's own sperms as in foreign particles, and it will produce anti-sperm antibodies. And also it can be seen even in the in traumas which we can see in lens crystalline. So in that case also we can see the sequestration. So sequestered antigen. So what are the proposed mechanisms? Genetic abnormalities or mutations, sequestered antigen. That means it is a normal tissue, but when it undergoes sequestration, it will be considered as a foreign particle, that is sequestered antigen. Loss of tolerance, loss of immune regulation, cross-reactive or shared antigens, altered forms of self-antigens or neoantigens. So these are the proposed mechanisms for the induction of autoimmunity. So sequestered antigen or hidden damage. So first thing. So when the antigen is released from the hidden location is nothing but many self antigens, like our body's own tissues, like own self antigens are hidden in some locations like CNS, testis, eye, cornea. So these will be like hidden forms. So when there is an organ damage, as I said previously, when there is an organ damage, at that time what will happen? Whatever the hidden antigen which is there, that will be released. So when it is released, it reaches the bloodstream. That means it will enter into the circulation. So once it is in the circulation, so this antigen, as it is present there, our body will produce some immune cells in order to identify it or in order to fight against that. So there will be encounter of antigen sensitive cells and stimulates this autoimmunity. Is that clear? So in our body, there are some self or hidden antigens out there in certain parts like CNS, testis, and eye. So what will happen when there is a damage or injury to that particular organ in which the hidden antigens are there? So once there is an organ damage, whatever the hidden antigen is there, that will be released. So So when it is released, it will enter into the blood circulation. So once it is in the circulation, it will be sensitized, and then it will stimulate the autoimmune reactions. So this is all about sequestered antigen or hidden antigen. And then alternate forms of self-antigens or neoantigens. Neoantigens, neo means new. That means newly forming antigens. So if you see that physical irradiation, like if there is if there is an exposure to radiations and chemicals like to exposure to certain drugs and biological things like viral infections. So when these three things are there, what will they do? They will act on the native tissue antigens. So radiation, drugs, or viral infections, or bacterial infections, whatever it may be. So when they comes in contact, or when they attacks on this tissue antigens like normal antigens. So in the presence of these physical, chemical, or biological factors, they will be undergoing these mutations. So once they undergo mutations, they will be converted into new neoantigens. So generally they are self antigens. Self antigens are nothing but self proteins. So once they undergo radiation, drug reactions, or viral infections, they will undergo some certain mutations. So because of that, there will be alterations in the proteinic structure. So that we call it as upon an antigenic alteration. Because of these factors, there will be alterations in the protein structure and will produce a new thing, a new protein that we call it as neoantigen. So when there is formation of neoantigen, again our body will start production of antibodies against to that and causes cell injury. So this is how it causes autoimmunity, altered form of self-antigens or neoantigens. And then genetic factors. It's like genetic factors like are also involved in these autoimmune reactions. So there is increased expression of class 2 HLA human. What is HLA? That you have to say. What is HLA? Human leukocyte antibody. These are the antigens on tissues involved in autoimmunity. Class 1 MHC. MHC is major histocompatibility complex. So these are the things that are involved in genetical factors. And there is increased familial incidence of some of the autoimmune disorders. And microbial factors include infection with any microbes like viruses, bacteria, and also mycoplasma, and viruses like Epstein-Barr virus infection, bacterial like Streptococcus, Klebsiella, and mycoplasma. So these are also involved in these autoimmune diseases. And then other is cross-reactive antigen or molecular mimicry. Cross-reactive antigens or molecular mimicry. So sharing of epitopes between an infectious agent and its host. So even on the infectious agent and the host, there is a non-proteinic thing that we call it as epitopes. So when there is an sharing of these epitopes, antibodies will be directed against the infectious agent, starts reacting with normal self-antigen. So when infectious agent and host both are there, they will share this epitope, that is non-proteinous molecule. So both of them will be having the same kind of thing. So whatever the antibodies that will be produced, that will not only act on this infectious agent, but also will act on the host. So thereby it also triggers the autoimmunity. And then mechanism of cross-reacting. Some organisms carry antigenic determinants like microbes. These antigens resemble host cell components known as cross-reacting antigens. So whatever the molecule or whatever the protein which is similar in both the antigen or the microbe and the host tissue, both of them, then we call it as cross-reacting antigens. So it will induce immune responses. So when there is any entry of any foreign particle, it identifies it by sensitization, and it also identifies the same molecule which is present on the host tissue and produces antibodies against both the antigen and also the self tissues, thereby induces immune responses causing the damage of organ or tissue in the host. And loss of immune regulation. So immune regulation will be done by lymphoid cells like plasma cells, B cells, and T cells. So because of any defect or lack of efficiency on these lymphoid cells, it results in the loss of immune regulation, leading to loss of tolerance control. So loss of immune regulation, it causes loss of tolerance. Loss of tolerance is nothing but cause of autoimmunity. And then lymphocytes abnormalities. Lymphocytes abnormalities include what are the different lymphocytes, B lymphocytes and T lymphocytes. So primary abnormalities either in maybe in B cells or T cells. So because of the abnormalities, proper immune regulation process will not be taken place, causing the more amount of immune reactions. So abnormalities in lymphocytes could affect any one of the mechanisms that normally maintains self-tolerance. So because of these abnormalities in any of these B cells or T cells, it causes a loss of normal functioning of immune tolerance. So these are the mechanisms by which autoimmune reactions takes place. And then types and examples of autoimmune diseases. So depending upon the antibody formation, these were classified into two groups. They are like organ specific diseases and organ non-specific diseases. Like in organ specific diseases, whatever the autoantibodies that are produced, that will react specifically to a particular organ or the particular tissue component and cause its chronic inflammatory destruction. So organ specific diseases, that means autoantibodies which are produced that will specifically bind to a particular organ of the tissue, and thereby it causes chronic inflammatory destruction. So what are the tissues that are affected? Are endocrine glands, alimentary tract, blood cells, and other tissues and organs are involved. And then organ non-specific diseases, that means the antibodies which are produced, they will not react only to a particular organ. They may react to any antigens, similar antigens which may be present in any part of the body, and causes systemic lesions. And in this condition they will form systemic lesions in organ specific. It causes chronic inflammatory destruction. So here is a table showing autoimmune diseases. So organ non-specific or systemic autoimmune diseases, it includes systemic lupus erythematosus, rheumatoid arthritis, scleroderma, polymyositis, dermatomyositis, polyarteria nodosa, Sjogren syndrome, Reiter's syndrome, Wegener's granulomatosis. In all these conditions, there will be formation of lesions can be seen. And then organ specific or localized, that is to a particular organ. It includes endocrine glands, it includes Hashimoto's thyroiditis, Graves' disease, type 1 diabetes mellitus, idiopathic Addison's disease. And alimentary tract, in that autoimmune atrophic gastritis in pernicious anemia, ulcerative colitis, and Crohn's disease. So these are the autoimmune diseases related to alimentary tract or GI tract, gastrointestinal tract. And then blood cells. In blood cells, autoimmune hemolytic anemia, that means own red blood cells will be broken down, and autoimmune thrombocytopenia, that means decrease in the platelet count. Because of these autoimmune reactions, it will damage or it will cause destruction of thrombocytes or platelets, causing decrease in the platelet count. And pernicious anemia, vitamin B12 deficiency anemia. And then others like myasthenia gravis, autoimmune orchitis, autoimmune encephalomyelitis, Goodpasture's syndrome, primary biliary cirrhosis. So these are the diseases which comes under autoimmune diseases. And also we have seen what like organ specific and non-organ non-specific. So this is all about autoimmunity. And thank you.